Hello everyone, my name is Muhammad Aquib, group number 322, and today's my topic is chemical aspects of enzyme action. Means how enzymes are made and how enzymes are works. The characteristic feature of enzymatic catalyst are the high activity and strict specificity with respect to the substrate or a group of substance, and the molecular activity of turnover number of an enzyme is defined as the number of moles of substance converted as per minute by 1 mole of enzyme at a considerable excess of substrate. So substrate is more in enzyme, substrate is more and enzyme is less, and substrate is converted into the product with the help of enzyme does interfere with the substrate. How fastly or how it means slower it change into the product, that is the main uh, uh, work of enzyme, how the enzyme is catalyze the reaction, and reaction will be fast. An enzyme never used in the reaction, it just act as the mediator and just does the work. So the properties of the enzyme are studied in vitro. Many enzymes have been obtained in crystalline form. Means we get the enzyme in crystalline form, just like a crystal. It substances are small molecular or small group of large molecules. On the contrary, enzymes are macromolecular substance. Means their weight is more. So basically, these are proteins except ribosome. All are the most of the enzymes are the proteins we know. Hence, the substrate interacts directly with the definite small portion of enzyme called the active site. Means the active site of that enzyme where the substrate get binds, and that substance change into the conformational change occurs in that substance, change into the product. The nature of the active site, that is the amino acid composition and sequence. Means and there is a special amino acid sequence that makes that active site, the nature of the active site, and also the cofactors, the inter its composition has been stabilized for a number of enzymes. We have already mentioned the enzyme substrate recognition, the changes in activity that arises as a result of chemical magnification of protein. It will possibly to reveal the functional group of the active site. Information on its structure is provided by optical and spectral mm, method, and also by the X-ray structural analysis of complexes of enzyme with competitive inhibitors. Thank you. And my second topic is conformational properties of enzyme. In order to understand enzymatic activity, it is necessary to consider the conformational behavior of the protein macromolecules. The conformational lability of protein makes possibly its interaction with substrate and other ligands. In some conformations of proper proteins bind the substrate more effectively. Simultaneously, there takes place selection of conformational changes of the substrate. The conformation of the protein and substance mm, that are selected into the enzyme substrate complex means enzyme when bind with the substrate, it forms enzyme substrate complex, and there will be substrate, in the substrate there will be conformational change occur, and that substrate change into the product, and the enzyme get off the reaction. Enzyme never used in that reaction. Thank you.